So it's my honor today, I want to keep this very brief, but I represent the Center for Redox Biology and Medicine as co-director along with Christina Ferdui. It's my honor to introduce Kelvin Davies, who will speak to us as our keynote speaker this afternoon. He was born and raised in London and is a dual citizen of Great Britain and the USA. His academic training has been primarily at the University of Wisconsin, University of California at Berkeley, and Harvard. He was first a faculty member at Harvard, then Albany Medical College in New York, before in 1996 joining the Andrus Gerontology Center at the University of Southern California, where he holds an endowed chair and serves as director. He's also Dean of Faculty and Dean of Research at the Leonard Davis School of Gerontology. And he directs a PhD program in biology of aging between the USC and the Buck Institute for research on aging. His accomplishments and honors are many. I could go on, but I'll name just a few. He's the founding editor-in-chief in 1985 of a premier journal in our field, Free Radical Biology and Medicine. He still holds that role, so many years. He also has past and present roles as editor-in-chief, as well as associate editor of 6 other journals. Important to note is that he was knighted in 2012 by the President of France into the National Order of the Merit of France. He's a fellow of the Society for Redox Biology and Medicine, the Gerontological Society of America, and 6 other royal societies and colleges. His scientific work in oxidative stress and adaptation has led to the publication of over 200 peer-reviewed articles, 2 full-length books, and many others. He's also, I note, held leadership roles in the arts. Most notably, I was impressed that he's past president and current board member, the California Philharmonic Foundation and California Philharmonic Orchestra. So let's extend a warm welcome to Sir Davies.
Good afternoon, everybody. Well, that was incredibly embarrassing, Leslie. I thought you were joking last night when you said you would do that. It's traditional at this sort of point in the proceedings to thank the organizers for inviting you. I'm going to actually tell the organizers I'm really cheesed off at them. Imagine following this young man, right? So bear with me. I'm not going to be half as good as he is, but I'll try to do my best. I also have an apology for you, because I thought there would be a huge screen up behind me. So some of my slides have words that are much too small for these little screens, and I apologize for that, but we'll try to do our best and see how we can work with it. So I'm at the Leonard Davis School of Gerontology, which is housed in the Ethel Percy Andrus Gerontology building. Some of you may know I saw an AARP table out here. Where are the AARP people? There they are. So I had dinner last week with Ms. English, your previous director, and some of you may know that the Leonard Davis School of Gerontology is named after Leonard Davis. That's pretty obvious. And Ethel Percy Andrus, the 2 of them got together and actually created AARP many years ago, and our building was partially built by AARP as well. So thank you, AARP. Yeah, that deserves a round of applause. You're right. So we all know what aging looks like, we think, right? This is pretty obvious. You go around from the bottom, going around, and you can tell right away what age people are, except you really can't, because lots of us spend our whole time really trying to figure out what aging is, how it works, why it works, and in some cases what you might try to do about it. As far as we know, this is the longest lived human being. She lived for 122 years. That's been carefully recorded, and we have the facts on that. There are a lot of other people with longer claims, but as far as we know, it's not proven. So we know what aging looks like, yeah? Well, this is a 70-year-old grandmother. And this is what astronauts look like these days. So our whole concepts of aging are changing. They've been changing for some time, but in some ways they're changing quite drastically. So I'm going to talk about a free radical approach to aging, and you've all seen things about free radicals. It's hard to walk out of a supermarket checkout counter without reading something about free radicals and vitamins or antioxidants. The subline is why we all get rusty, like this car is. This is the Queen Mary 2 coming into San Francisco Harbor through Golden Gate Bridge. They actually had to lower the funnel to get her through, and that was planned in the building. They knew exactly the size, and it only works at low tide, so she can only get out at low tide too. But eventually, even the Queen Mary 2, as grand an old lady as she is, or young lady, excuse me, she's a young lady now, she will eventually rust like this. So why do we all get rusty? It's the oxygen. And I'm going to talk to you quite a bit about oxygen. So oxygen is silent, invisible, no smell, and it supports combustion. Things burn because of oxygen. I apologize for this being small. So it's a very toxic gas. It's an extreme fire hazard. Exposure to 100% oxygen will kill you after a few hours. At 20% oxygen, we jokingly say the toxic effect is delayed, and it takes maybe 80, 90 years before it kills you. Because it contributes to a complex process we call aging. It's habit forming. One breath at birth and you're hooked for life. It's the ultimate drug. Whoops, went back. It's a fire hazard, we all know that. If oxygen were invented today, your government would have to ban it because it's far too dangerous to be allowed. So where did it come from? Why is it dangerous? It came from photosynthesis. That is to say, it was released into our atmosphere because of photosynthesis. Obviously, it's an element and it exists in its own right, and it's a strong oxidant. So it came from these sorts of things, green leaves, these other pretty things, and these. But in fact, when we think of photosynthesis and we think of releasing oxygen, we think of large plants and the like. But in fact, most of the oxygen that's been released and still being released into the atmosphere comes from blue-green algae or cyanobacteria in the oceans. And they're not all blue-green. Some of them are other colors too. So these things far outweigh, actually, in oxygen production, the terrestrial plants. But nevertheless, they're all important. So watch out for the oceans. They count. I'm a sailor, so I really care about the oceans. It's the waste product of photosynthesis is how it comes about. So carbon dioxide plus water with little light and energy gives us glucose. That's the basic building block for plants to grow and for everything to grow, plus oxygen as a waste product given off. So plants were some of the first toxic waste dumpers on this earth, or rather the blue-green algae were, and so the plants followed along. So some of you, some of your children, my children, saw this in grade school as the cycle of life, so to speak, one of the many cycles. Photosynthesis for producing oxygen, we respire on the oxygen, we give carbon dioxide off, and the plants use the carbon dioxide to build themselves. It's a nice story, and it actually does happen. The problem is we've kicked the whole thing way out of balance by producing way too much carbon dioxide. But that's another story for a different day. So how do we get oxygen in the Earth? If you look back, this is a scale along the bottom of 4.5 billion years, as far as we know the story of Earth. And about 2.5 billion years in, oxygen started to appear in the environment, as far as we can tell from the geological record. It wasn't there very much before, and it started to come about because of all these blue-green algae and then terrestrial plants releasing oxygen, dumping oxygen into our environment. It's a little more complicated than that, but that's good enough. Who discovered it? Well, it depends where you live. If you live in Great Britain, where I was born, then it's Joseph Priestley. If you live in France, it was Lavoisier. And if you live in Sweden, it was Schiele. Actually, I'll leave the dates to you, but if you can read them, 1733 is the old, anyway. Right, you get the point, thank you. Can I pay you, or did I already do it? Thank you. So Priestley actually was even cleverer than that, or prescient, one might say. He called dephlogistered air, phlogiston was what he thought he was dealing with, and actually that would be nitrogen, because what he was doing was removing it from air. What you would be left with then is largely oxygen. And he said, I'm so used to using a pointer that I keep pointing with this, that dephlogistered air, in other words oxygen, might be useful as a medicine. It might not be so proper for us in the usually healthy state of the body, for as a candle burns out much faster in oxygen than in common air, so we might, as it be said, live out too fast and the animal powers be too soon exhausted in this pure kind of air. So he was actually probably the first person to make a link between oxygen, although we didn't know it was oxygen then, and early death. Following a long time afterwards, Rebecca Gershman and Dan Gilbert proposed that the harmful effects of ionizing radiation could be ascribed to oxygen free radicals. And then Denham-Harman argued that the pace of aging could be related to the level of free radicals produced by metals or respiratory complexes and cause damage to tissues. And I'm going to sort of progress from there. So what are free radicals? I've been mentioning them a few times, but let's define them. Free radical is any atom or molecule with one or more unpaired electrons. It's a nice chemical definition. And they're reactive because they try to steal or donate their extra electrons. So oxidation is the process of losing electrons. If you give up an electron, you've been oxidized. The thing that did it took the electron. It became reduced in the process. It is an oxidant. So those are our definitions. And the net effect of free radicals is usually chain reactions, so many, many molecules can get damaged one after another. And there's a term called oxidative stress, which implies that tissues can be overwhelmed by oxidation and severely damaged by it. Chemists knew that, and physicists knew that free radicals were important in all kinds of chemical reactions over the years, but biologists didn't really believe it for a long time. The big change really came in 1969 when Joe McCord and Erwin Friedrich found an enzyme. So this is a protein whose manufacture is encoded in our DNA. It's a gene product that actually its only function in life is to remove an oxygen-free radical called superoxide. You take oxygen, add one extra electron, and you've got superoxide, which is an oxygen-free radical. I apologize to all of you. My back's to you all the time, but I can't read the things otherwise. I'll try to, yeah. Here I go again. So aerobic, as I said, I'm used to the screen. Aerobic respiration. Why do we need oxygen? What's it for? Why do we need it for us? Why do all animals that we know of use oxygen? And we use it for respiration. We transport oxygen to tissues, and in the tissues, in our tissues, we use it to oxidize foodstuffs. In particular, though the simplest form, your brain works on glucose. If you use a simple form of energy production, which is called glycolysis, it's the sort of simplest method we have of getting energy from food, you get a little bit of energy. If you fully oxidize that glucose with oxygen in your tissues, you get 18 times more energy from the same molecule of glucose. This process actually enabled multicellular organisms to develop and ultimately led us to develop. This occurs in a part of the cell called the mitochondria, and you heard a talk earlier about mitochondria and how they're involved in production of energy in cells. I'll come back to that. Problem is that oxidation is often incomplete, and there are partial oxidation reduction products, in other words, free radicals, either stealing or donating an extra electron. So here's a cell, this nice big round thing, obviously looks nothing like a real cell. How do they get the energy to live? Well, those, you can just about see in the bottom left-hand corner there something that says mitochondrion. Hopefully everybody can see that. That's the part of the cell that makes the energy. It turns out that mitochondria actually originally were bacteria, and at some point in our ancestral past, bacteria became symbiotic with the simple cell, and now the two coexist. So you have mitochondrial DNA as well as your chromosomal DNA, and all of your mitochondrial DNA is inherited only from your mother. Your chromosomal DNA is split parentally, but mitochondrial DNA is maternally inherited. So mothers rule in this one. So mitochondria are called the powerhouse of the cell. Here's a powerhouse. If you notice, there's a sort of a single membrane all the way around, and then a membrane that invaginates back and forth and back and forth and back and forth. And that's a common theme in biology. It's a way of packing a lot of things into a small space, like a concertina. And packed into that small space membrane are lots of proteins, and those proteins conduct the process of oxidation of foods and consumption of oxygen to oxidize those foods and production of energy. And you heard it called ATP earlier, and that's what it is. Up at the top right, you can see a cross-section through skeletal muscles, so any of your muscles in your body, for example. And the dense bodies, if you see the lines, there's 3 vertical lines, big dark ones. On either side of them, there are mitochondria. And so mitochondria are packed very tightly in your muscles to provide lots of energy for contraction. Here then we get at the top of the slide, the outer membrane of mitochondria. In the middle of the slide, this invaginated inner membrane that's full of proteins that are involved in production of ATP and production of chemical energy from foodstuffs. And there's two things there that look like O2 and a minus, O2 and a minus. That's that superoxide radical I was talking about earlier, a free radical that's produced accidentally, as it happens, because the process isn't perfect. It's actually about 95% efficient, which if you compare it with an internal combustion engine in your car, if anybody's using gasoline, petrol, it's much better than you get from your car. But 95% is not 100, and there's some leakage of electrons. So a major source of free radicals in our bodies is from metabolism, it's from mitochondria, but there are many other sources. Air pollution, a major one. Cigarette smoking, it's partial combustion. Lots of free radicals. Ionizing radiation, if you're exposed to gamma rays, or if you're an astronaut and you get exposed to cosmic rays, you're in danger. Ultraviolet light, too much time on the beach, photooxidation is a major source of damage. And inflammation, many forms of inflammation are producing free radicals, producing damage to your tissues unintentionally. So again, multiple sources, radiation, partial combustion, metabolic sources, medical and dental x-rays, which is why we worry about them having too much of an exposure. And we actually have cells in our bodies, if you look in the middle where it says metabolic, phagocytes, neutrophils, macrophages, monocytes, glia. These are cells in our body that are trying to kill off invading organisms. So if you get a bacterium that's trying to colonize you, you don't want that to happen, you want to kill off those bacteria or at least stop them from growing. And the way we do that is to produce free radicals that cause damage to those bacteria or kill them off, and then we basically eat them and use their proteins. So the major free radicals in biology are partial reduction products of oxygen, and most of the oxidative stress in animals and humans is caused by oxygen free radicals. So here's a little bit of chemistry. You don't have to worry about it too much. There will not be a test later on. So superoxide, oxygen is reduced by one electron to superoxide. In the middle there you get hydrogen peroxide that's produced in our bodies. Up the top is hypochlorous acid. Anybody know what another name for hypochlorous acid or hypochlorite might be? Thank you. Common or garden bleach. So these two things that you can buy in your supermarket, Clorox or hydrogen peroxide, we actually make in our own bodies, and we make pretty good amounts of them too. But we use them in order to kill off bacteria. Photochemical smog is a major problem, lots of partial reduction products. These are some of the oxides of nitrogen that are in smog. A small percentage of what's in smog. We won't go through them all, it's crazy, but there are thousands and thousands of partial combustion products. So this led me to something I call the oxygen paradox, excuse me, which is the idea that oxygen is necessary for life, yet it's toxic to us. And in fact, that's a paper on the left that I wrote in 1985, and a book I published in 1995 both with the same title of the oxygen paradox. So, which has intrigued me for years. Now let's go back a little bit, because I'm at the point where I'm sensing, okay, he's given us too much chemistry and all of these things. So let's take a little lighter look. I started a lot of these studies when I was at Berkeley. This is the University of California at Berkeley, where David and I both were. That's Sadler Tower on the left, and over on the right is doing what a lot of us did back in Berkeley in the 70s, which was to complain. But imagine now I was trying to study free radicals and talk about free radicals in the early 70s at Berkeley. When anybody remember this guy? Yeah. Timothy Leary? Yeah. We don't need to say any more because we're old enough to, right. So the politics of ecstasy. Timothy was an interesting guy. And of course, at the same time, across the other side of the Atlantic, there was a little country called the Soviet Union, or a series of countries, that had free radicals they liked a lot. That was a good thing. So I started this journal called Free Radical Biology and Medicine, which was alluded to by Leslie, and we wanted an international advisory board. So I found myself calling people in the middle of the night in various parts of the world, including Russia, including Poland, various other places, asking them to join our international advisory. I know my phone was bugged for quite a while. So anyway. So how do we defend ourselves against oxidation and survive in this toxic environment? How do we deal with the oxygen paradox? Well, there are some good antioxidants that we can use. Somebody was asking me before about vitamin C. There's vitamin E that's in your membranes, beta-carotene, and all sorts of other things. Oops. These largely come from there. If it's brightly colored and pretty, it probably has a good source of antioxidants in it. Or some of these, very good source of antioxidants. Problem is, they're designed to protect plants or fruits from photooxidative damage. They weren't designed to protect us. So the idea that we assume that they will protect us is not necessarily good. Secondly, can you imagine how much of, for example, let's go back one. So look at the yellow fruits on the right, or the tomatoes. Think of how much lycopene you actually need, or beta-carotene, to get that much color throughout your whole self. That's what you need to protect yourself effectively against photooxidative damage. I don't see anybody around here who wants to look one of these colors. Well, there's already one in the White House, so. But, thank you for that. This is one of my best slides, not to say that it's great, but it's decent. If you look carefully, you'll see a drip going on. So there's a lemon or an orange, and it's dripping on this sliced open apple. And if you, on the side where there's no dripping, it goes brown, it oxidizes. And on the side where vitamin C is dropping down all the time, it stops from being oxidized. Now, this makes a nice story. Unfortunately, I have to tell you it's only partially true. It's also partially preventing the browning reaction, because the lemon juice or orange juice is very acidic, and in part it's the acidity that prevents the browning. But nevertheless, it makes a point that vitamin C is a good antioxidant, but you need a lot of it in order to be effective. And most of the experiments that have been done over the years are done in test tubes, where you can put a lot of vitamin C protecting a little bit of oxidation. So this works, but it works only in very large amounts. On the other hand, that hasn't stopped all of your favorite magazines from telling us how important all these things are and how much we should take. So this is the checkout counter sort of stuff that you'll see. But is that what you want breakfast to look like? Not mine. Lipton's also is in on the act, not the only one. Lipton's unique combination of health benefits with antioxidants to fight free radicals. There's also, I saw the other day an ad on television. I can't remember what it was called now, but there's sort of soldiers fighting with, what's it? Pomegranate. Yeah, quite amazing. Yeah, I won't say any more. So there's something else called the French paradox that many of you may have heard of. It was the idea that French people eat lots of fatty foods, and they do all kinds of other things that you shouldn't do, and they smoke, and yet they don't die earlier than we do, or than you do, of heart attacks. And maybe it was the wine. And that's become a very interesting topic. There's all kinds of ways to look at that. In fact, a couple of years ago, my wife and I organized a conference in LA with lots of French people. And in fact, we all got together and wrote this ridiculous paper with too many authors, where we looked at the oxygen paradox, the French paradox, and age-related diseases. And that's available online if anybody wants to see it. It's freely available. You don't have to pay a penny. My question is whether my favorite martini gives me the same sort of benefit, but we haven't figured that one out yet. So you get this idea, or people have built up this idea, that there's sort of tug-of-war going on between the free radicals and the antioxidants, and one's pushing and pulling and who will win. Or this one, this sort of medieval Castle idea of antioxidant defense. So down the bottom closest to me, you have the nasty oxidants attacking, and then you have this line of free radical protectors defending the castle. My view is, why wouldn't they have stayed inside the castle and just thrown rocks or something? But anyway, the big deal here is that most dietary antioxidants work to some extent, but you would need so much of them to be completely effective that you'd all be green or yellow or red or purple or whatever. You can't take that much in. It's just simply not possible. So we do have other things. We have antioxidant enzymes, and that's where I started with the superoxide dismutase that Erwin Friedrich and Joe McCord found. So we have actual enzymes whose role in life is to get rid of free radicals, and they do a very good job. They actually do much more than the antioxidants do, and we make them all the time, and we make them ourselves. On the other hand, despite the antioxidants, despite the antioxidant enzymes, damage to proteins, lipids, and DNA still occurs. Despite all our best intentions, it still occurs. So what do we do about it? Could we remove the damage? Could we repair the damage? And actually, we do a little bit of both. So I'll tell you a little bit of some work that we do. There are actually enzymes that work on damaged lipids, damaged DNA, and damaged proteins. We started with proteins, and with the idea that I told you there was going to be a light bulb in the anyway, so there are a couple of enzymes that really are very good at removing damaged proteins from cells. One of them is this thing called the 20S proteasome that has different enzymatic activities. And another one is the LOM protease. The 20S proteasome is present throughout your cells, everywhere in your cells, except inside those pesky mitochondria that don't have proteasome. They have LON instead. And you can see there's a sort of similarity. Things have to get, so a damaged protein, that's the mitochondrial matrix, a damaged protein gets pulled in to one end, and then it gets degraded into little chunks that are called amino acids and small peptides, and you can reutilize most of those. So that removes the damaged protein. If you don't do that quickly, it turns out most damaged proteins start aggregating, cross-linking, and becoming permanent parts of your cell and then causing further damage. So it's important to get rid of them quickly. I know this is impossible, sorry. Again, big screen needed. But basically what it says, if you start out on the left, lots of sources of oxidation, trying to convert normal proteins, lipids, and DNA in your cells into damaged constituents. And then there are things that are trying to prevent that. So the antioxidant enzymes, the antioxidant compounds are trying to prevent that. And then just down here, this damage removal bit that it says there for proteins, for lipids, for DNA, removing damaged portions and trying to repair some of the damage that's done. So that's a lot of, really, when you think about it, a lot of systems to cope with oxidative stress. And as we said, nevertheless, damage still occurs. And this free radical theory of aging has proposed that oxidative damage to proteins, lipids, and DNA accumulates over time, eventually causing senescence. That's then Harman's original theory. We've sort of worked on that quite a lot over the years. This is a paper back from '91, so it's not current at all, looking at how these damage removal systems might be involved in protecting you against this. But again, is that all there really is? Or is there even more? Is there another level of protection? After all, this is a pervasive problem. We're living in an toxic oxygen environment. We really have to try to find ways to deal with it. And it turns out that, so if you take a normal cell at the top, trying to be converted by oxidants to an oxidatively damaged red cell at the bottom and then perhaps dying, you have what's preventing that? You have the antioxidant enzymes and compounds on the far left. Let's miss the transient growth there. There's some direct repair. There's damage removal and replacement or repair. And then far over on the right, there's something that says adaptive responses, which is a lot of what my lab works on. So adaptation to oxidative stress, it's a phenomenon that my lab calls adaptive homeostasis. And we'll try to talk about that a little bit. So far, I've sort of presented all of these defenses as being static things. They're there, they do their job, that's it, no change. But in fact, we can show that in any living organism, from a bacterium through yeast to simple cell organisms to mammalian cells to worms to flies, let's see, what else have we studied? Sea urchins, mussels, oysters, you name it, we've done a lot of them. They can all adapt transiently, short term, and increase their defenses by in many cases increasing proteasome and LON, those two proteases I talked to you about that degrade or remove damaged proteins. DNA repair is also involved, lipid repair is also involved, but you can only study so many things. So my lab tends to focus on the proteolysis side, the protein degrading side of things. So this is adaptation to oxidative stress to alter gene expression. We'll skip this one. Here's a simple experiment that sort of gives you the idea of what I'm talking about. So up on the top, way top on the left, these are yeasts, by the way. I can show you the same experiment with mammalian cells, but it turns out when you study mammalian cells, you use a blue dye. It doesn't show very well in lighted buildings. Yeasts show up really well. Control cells on the left on a plate, nothing done to them. Bottom left, cells challenged with hydrogen peroxide, one of those oxidation products we talked about. And you see you lose more than half the cells. Top right-hand corner, cells treated with a very, very low concentration of hydrogen peroxide. So maybe 1,000th of the concentration that was used in the bottom left. And then you wait about 45 minutes to an hour, and then the bottom right, treat them again now with the high concentration of peroxide, and you see they all survive. So these cells in about an hour have gained the ability to resist this oxidation and damage and death, in fact, from hydrogen peroxide. And it turns out that that resistance lasts in yeast, it lasts for a couple of hours and then it goes away. If you do it in mammalian cells or human cells, it lasts for about 36 hours and then it goes away. And you can actually cycle through adaptation, deadaptation, adaptation, deadaptation. So it's an important phenomenon. It occurs in everything we've ever looked at. If you get on the freeway and you're exposed to lots of smog, you will start adapting to it. If you go down to a shipyard and they're doing arc welding, lots of ozone, you'll adapt to the ozone. And you de-adapt over a period of hours too. And over 100 genes are the expression of over 100 genes is upregulated. In other words, you make more of their protein products. Actually, a similar number is downregulated. So it's a massive change in gene expression. All right, what's going on? This is a process, or mimicking a process we call homeostasis. This is sort of the backbone of modern day physiology. It's the idea that, let's make it straightforward. If you go to your physician and they take your blood pressure, you don't expect them to say, or they take your heart rate. They're not going to say, you should have this number. They're going to say, you should be between here and here. That's the range, the normal range. Well, didn't mean to do that. That's the normal range shown here. And it goes up and down. There's diurnal variation. There's all sorts of other reasons to change. If you take your blood pressure first thing in the morning, it'll be slightly different than it would be in the evening. We go up and down within a normal range, you hope. What we're seeing then is an expansion of that range. Adaptive response is either expansion or contraction. Actually, it can work both ways. And so we call that adaptive homeostasis. Okay. And here, looking at the same sort of thing, here's your heart rate within a normal range. With adaptive homeostasis, you get a signaling molecule like hydrogen peroxide in our case that increases the range, turns on signaling pathways. We won't get into all of what those are, but there are discrete pathways that signal the increased production of gene products for all of these 100 genes more and more. Adaptive responses are turned on, so proteasome and LON are being made at much higher rates than usual, 2 of the 100 or more gene products. And then after a period of time, that response is turned off and you return to the basal range. So adaptive homeostasis now has stopped and you're back to normal homeostasis. Well, it turns out, so all is good, right? It turns out that in aging, this doesn't work quite so well. And this is part of the aging, we think part of the mechanism of how aging works. So diminished adaptive homeostasis of proteasome and LON, they contribute to aging processes. These are some experiments we did a long time ago, well, in scientific terms, back in 2000, seems like a century ago, where we were looking at increased damage to cells and increased protein damage to cells, accumulated damaged protein with different kinds of cellular aging. And in fact, if you look at animal and human studies, real studies of real people, as well as real animals, there's a clear accumulation of oxidized and cross-linked proteins with age. So something is going wrong, something is not clearing all of those damaged proteins as rapidly as it should when you're old as when you're young. And this is part of the reason. So if you look at we've got homeostasis there between the 2 dotted lines, normal homeostatic range. If you look at the top curve, that's the young curve, that's the range you've got for adaptive homeostasis. The middle age curve is a bit lower, the age curve is lower still, and also the baseline shifts. So adaptive homeostasis range diminishes, and the baseline, or the basal range, compresses. And we've just, this is from a paper we published just the end of last year, and 2 papers in the Journal of Physiology and then Clinical Science. These are all open access. Everybody can go and find these for free. And looking at many stress exposures. I've been talking to you today about oxidative stress and about oxygen radicals and about the oxygen paradox. But in fact, these papers we pulled together work on oxidative stress, heat stress, cold stress, metabolic stress, environmental stresses, hypoxia, exercise stress, so it goes on. It's a bit boring list, but lots, even including emotional and psychological stresses, which do have some effects on your physiology as well as your mental state. And in every case, we see that the young are able to adapt very well of every species we've looked at, whereas the old are much less able to adapt. So there's a loss of adaptive homeostasis that we think makes you much more susceptible to damage and makes you less able to remove damaged products when they're being made. So we sort of revised this free radical theory of aging. We say oxidative damage to proteins, lipids, and DNA accumulates and or increases over time, contributing to senescence, may be caused by diminishing mitochondrial function and increasing electron leakage, so more damage occurring. By inhibition of damage removal or repair enzymes, I didn't show you much about that, but many of the damage removal and repair enzymes like proteasome actually become less functional over time, and or by diminishing capacity for adaptive homeostasis. Now anybody know what this is? No artists in the group. It's the fountain of youth, and we haven't yet found it. Sorry, but not today. On the other hand, we're hoping, some people are hoping for this. Take 2 of these pills and call me a century from now. Not going to happen. Bad news for you, sorry. Unless you're a Reader's Digest fan, which has already published that there's a new pill that ends aging, so it's already done. I can go home and hang up my lab coat. I don't believe that either. Benjamin Franklin said to lengthen thy life, lessen thy meals, which I should listen to. And just to finish, this is my lab group. These are the people who actually did the work. That's actually a free radical cake in USC colors. Don't forget who did that. These are the people who particularly contributed to what I told you about today. Laura Corrales Diaz-Pomato, Rachel Rains, Andrew Pickering, all former students, and collaborators Derek Seabuth, John Tower, Henry Foreman, and Tilman Grune. And I thank you all for your attention. And again, I apologize for having to read this, and I apologize especially to everybody on this side of the room to whom I was extremely rude. Sorry. Questions, if you have them.
Yes, have you looked into the, or your lab looked into the effects of plants or botanical extracts that upregulate NRF2?
Yeah, in fact, one of the slides I showed you had NRF2 on it, but I went through it
that.
very quickly. So the slide that showed adaptive homeostasis with proteasome and law at the end had working through signal transduction, NRF2 signal transduction pathway. We actually published a lot of papers on NRF2 signal transduction of proteasome and lawn. It's not the only, so this is the problem. Some of you in the audience are saying, what the heck is NRF2? And other people in the audience know all about NRF2. So I was trying to pitch a talk to everybody, which means, of course, I get nobody. But NRF2 is really important for proteasome, a little bit important for lawn. There are other systems that are upregulated that have nothing to do with NRF2 at all. So there are at least 8 or 9 signal transduction pathways involved in these responses. And I skipped over them.
Dr. Davis, I really don't.
I see you've got notes there.
I really didn't mean to ask a question because your talk was just perfect. But however, there are some botanicals that are known to increase NRF2.
Yes.
Okay.
Curcumin,
Yeah.
sulforaphane.
That's right. Are they useful in adaptive homeostasis?
So there are a number of products out there. I'm not going to suggest that you try any of them, because nobody really knows how well they work. Some of these things are found. Some of these things are found in cruciferous vegetables. So things like Brussels sprouts, for example, or good sources of some of these things.
But there are many, many beautiful compounds of fascinating pharmacology that activate NRF2.
Yeah. The problem is we don't really know very much about how well they work over any.
They do work.
Okay. I'm talking. They
do work.
They work in the sense that they can be shown to turn on certain signal transduction pathways.
Well, if somebody can't
excuse
walk,
me, listen to an answer.
Yeah, go ahead.
And then you can respond.
Sure.
We don't know over time how they may affect people if people use them long term. There's very little data on that, and there's very little safety data on any of that. So I would not advise anybody to go start dosing themselves up with these things. Sorry.
Well, the point I'm making is if somebody walking like this, okay, and you give him some molecule, okay, which is harmless, is present in every fruit that you can eat.
Yeah.
And then he starts walking straight.
Yeah, I love anecdotal stories.
Okay, and this is not a joke; it's a real story.
Yeah, but I'd rather deal with the verifiable science. Sorry.
No, it's based on science.
Where does the current fad of fasting fit in?
So yeah, there's a lot of work going on fasting, a lot of work going on caloric restriction, which has been going on for years, and now work going on what are called fasting mimicking diets. So these are diets that use, what people have noticed is that if you start fasting or you drastically calorically restrict, that the kinds of chemical changes that occur in your body might be mimicable by changing your diet strongly to certain kinds of foods and away from others. And people are working on this. There's all sorts of interesting results coming out. I don't work on it myself, so I can't speak strongly about it. But I have seen a lot of very encouraging results from some labs. Yeah.
I think we were referring to compounds like oleanolic acid, salvinolic acid, have been tested extensively in the National Library of Medicine, and they upregulate all your liver enzymes.
Yeah. You're saying it. Yeah.
It's on the National Library of Medicine.
Well, there's a lot of things
They've
in the
done
National Library of Medicine.
research. The research.
So I don't think anybody should take that as proven scientific way to avoid aging or diminish aging or whatever. No. Sorry.
So how does exercise fit in with the adaptive homeostasis?
So exercise also, actually exercise generates more free radicals and turns on NRF2 and all sorts of other systems. And clearly produces a beneficial effect if the exercise isn't too extreme. If it's too extreme, then you get bad effects. But many of the things you see with the kinds of effects that I'm showing you today are also mimicked by exercise. Not all of them, but many of them. And some of the things that are not done here. Yeah, I'm sorry, I don't mean to be too harsh. Since this is a generalized audience, I really want to stay away from giving health advice when things are partially understood,
Excuse
partially
me.
not.
I mean, we all have a hope for, as you said, the fountain of youth. So I commend you for your research and I commend him for his, but at the same token, you're here talking to us. So I have a question for you regarding possible, we said exercise. What about diet? Is a vegan a meal or the Mediterranean diet better for these radicals?
Not necessarily for dealing with oxidative stress. It might be more beneficial for other things. It might have some help with oxidative stress. Part of the problem is that a lot of the data that we have are data that was collected from survey studies. So we have lots of data, lots of epidemiological data, lots of post hoc studies, not done experimentally, not a priori, but done after the fact, and then you do a survey and ask people what they did, and then you collect all the data. And it's perfectly valid. It's just a question of how you do it. Those kind of studies are subject to all kinds of problems, not the least of which are people lying, and not the worst of which are people misinterpreting what they hear. So there's a famous study when people were looking at the French paradox, for example, about how much wine people should drink or shouldn't drink. Most of these data were collected by summer students going around and collecting data for their professors. So there's a famous story of young students, PhD students, going over to Sicily and collecting data from people in Sicily about how much red wine they drank every day. And they go off into the hills where there's a lot of sheep farmers way out, away from everybody else, alone in the hills with their sheep, and say, how many drinks would you have a day? And they say, well, 2, 3. And they go home, 2, 3 glasses. You know, these guys were talking about jugs that you could hold on your shoulder. So there's a little bit of discrepancy in some of that, and I'm making it fun. But there are real data out there. There are strong suggestions that the Mediterranean diet is more healthy for you than the hamburger diet, for example, to the point where, so that's I think good. Only some of it, I think, is probably working through free radicals. There's a lot of other reasons we age or go downhill. And then there are emerging data, as I said, on dietary restriction and on fasting or fasting mimicking diets that are starting to look really good. I don't think, we certainly don't have all the data yet. So those are promising areas. And there may be promising areas, there's a nod over here, there may be promising areas of certain plant products that might be used carefully and properly to stimulate things. But part of the problem we're seeing, what I didn't get time to tell you about, is that you saw that I put up slides that said in older animals, this adaptive process isn't working so well. And part of the reason that it isn't working so well is not because you don't have the apparatus or the equipment anymore, but because inhibitors are being produced, like BUC1, like c-MYC. There's a lot of work on this now, that both are antagonists for NRF2. So it doesn't almost matter how much you turn on an NRF2 system. If you have a lot of their antagonists being produced in old age, that's a problem. So there's a lot of work going on now to try to figure out why BUC1 is being produced as another protein in old age and what we might do about it. And again, that has a danger because it might be being produced for a real reason that's better than not having it. So it's baby steps all the time in this sort of thing. Yeah.
Is there a point where you lose the adaptive response with repeated challenges in the system?
Yeah, so we published a paper a few years ago looking at what happens if I said you can adapt and then de-adapt and adapt and de-adapt. It's true to an extent. If you bring the repeated bouts of exposure to an oxidant too close together, then you start to initially you get a super response. And then if you bring them too close, you start to lose the effect. You also lose the effect if the stress is there constantly. So that has all sorts of implications for the workplace. If you go to work, if you're exposed in the workplace all day, every day, but then you go home for 12 hours or more, is that enough time to de-adapt? When you go back again in the next morning, is that enough time to re-adapt? If you get exposed to a different thing in the area you live or on the freeway when you drive home than you were exposed to at work, is there cross-adaptation? So there's all sorts of real-life outcomes of this or questions about it that we've not answered, but that are raised by that kind of work. And there's a long, the EPA is now shut down, so we won't be doing very much work on that. But it'd be nice if they started something on that.
Sir Davis?
It's an important question.
Yes, Sir Davis.
Kelvin. Yeah.
Is the research meant to understand what's going on or to prolong life longer?
Okay, my research is I'm just trying to figure out what's going on.
Okay.
So I make no claims to know how to deal with it.
Because the question I was thinking of is you live longer, you don't understand the technology as well, your friends are gone, it's...
Well, we heard earlier that's the fallacy that you don't understand the technology, right? So
good point.
Friends being gone, yeah, we're all experiencing some of that. Yeah, I'm 67 and I've lost several friends actually recently, which is very sad. That wasn't happening in my 20s and 30s and 40s. So we all know how that feels. The technology, I think a lot of it is because we kind of shut ourselves off. I think a lot of times you get used to and comfortable with what you got. And people talked earlier about, and this is just my own opinion, people talked earlier about how lives are more diverse when you're younger and less diverse as you get older. And I think it's because you're making choices. You say, okay, I care about this. I've done that twice before in my life. I don't need to do it again. And I think in some ways you do that about technology as well. I know how to turn a light bulb on. I don't need to have a machine that I speak to that turns it on for me. Things like that.
What would you say about the ketonic diet?
I wouldn't.
You wouldn't?
No, I don't have an opinion. Okay. Sorry.
So I wish I could say I understood everything you said.
Me too. Did I fake it well enough?
But that
I
all
tried.
brings me to say I really appreciate your perseverance in the work that you're doing.
Well,
it doesn't matter that I don't understand it or that I will never use it in my daily life. But, at this stage in my life, I enjoy going places and hearing something that is so far away from what I normally encounter. it really makes me feel alive. So I really appreciate
Thank you.
it.
Just if I may, if I, as a disclaimer, in my own defense, sometimes some of the things that we do do have some benefit. So we're hoping, or I'm hoping, that a lot of the things we found will actually be used by people who are much more clinically oriented than I am, and would like to try to see how they fit into a clinical setting or what could be done with them. We actually did do something at one time that was very useful. There was a drug out there years ago, it's still being used, called adriamycin or doxorubicin. For a while it was being used only for children who had childhood leukemias, certain childhood leukemias. And it started to be called the Red Death. It's a pinkish red color because children were dying from it. And it turns out it's a dosage problem. The kids died of horrible cardiac problems. And I and a colleague actually showed how it happened, why it happened, what the mechanism of redox cycling of the drug, production of oxygen radicals, worse in the heart because the density of mitochondrial tissue is enormous there. And actually we figured out, and working with clinical colleagues, that if you just kept the dose low enough, you would avoid the problem. And now adriamycin is used, dosed carefully in all kinds of clinical chemotherapeutic settings. So every now and then. You know. Thank you. We good?
So let's thank Dr. Davies.
Thank you.
All right, and let's just thank all of our speakers for today one more time. All right, and so tomorrow morning, let me remind you, we start a little bit earlier. 8:45 is when the program starts. Breakfast again will start at 8 o'clock, and we hope to see you all tomorrow.